In the last video, we walked through the process of meiosis. And the reason that we are most interested in meiosis, or at least more so than mitosis, is that we are in genetics and we are interested in how DNA information is inherited or passed on to the next generation. So if we're talking about people, that happens through the fusion of gametes, or our sperm and egg cells. If we're talking about other things, plants, single-celled organisms that reproduce sexually, you still have gametes. You still have sperm and you still have an egg. Now, in humans, sperm are very different than the quote-unquote sperm or pollen in plants, and different organisms reproduce in different ways. But spermatogenesis, or the production of sperm, pollen grains from the males, are all going to occur through the process of meiosis, essentially unchanged from what we discussed last lecture or last video. Basically, meiosis is a continuous process. In humans, it occurs in the gonads, which are the sex cells that produce the sperm. And in all sexually producing organisms, they're going to have these specialized cells that undergo meiosis to produce the male sex cell. So in spermatogenesis, we get 4 similarly sized cells all at the same time. And this process is happening continuously throughout a male's lifetime. Basically, once he enters puberty, he continues to produce sperm throughout his lifetime. In females, that process is a little bit different. So in females, egg production is called oogenesis, and we'll look at this in a little bit more detail in lab. But instead of 4 cells that are produced at the end of meiosis, like in sperm production, oogenesis produces only 1 fully functioning cell. So this process actually spans across many years of a female's lifetime. So meiosis 1 is going to produce 1 large cell that's called the secondary oocyte, and a second smaller cell called a polar body. So if we start out with our primary oocyte, this is the cell that is going to undergo meiosis in order to produce an egg. It enters meiosis 1. And at the end of meiosis 1, it produces 2 cells, 1 large one and 1 small one. That large one is called the secondary oocyte. The smaller one is called a polar body. Now, in many organisms, that polar body doesn't even undergo meiosis 2. It basically disintegrates and the contents are absorbed by the female's body. In this secondary oocyte, we have a whole bunch of cytoplasm. It basically receives all the cytoplasm, or most of the cytoplasm, from that primary oocyte, and that polar body doesn't receive anything. However, that polar body does receive half the chromosomes, just like it would if it were undergoing regular meiosis. So half the chromosomes are in that secondary oocyte, half the chromosomes are in that polar body. Now remember, in meiosis 1, we are separating homologous chromosomes. Then that primary oocyte, once the female hits puberty, is going to undergo meiosis 2. And when that happens, we get the same thing where we get a large cell and a small polar body. So that polar body is again going to eventually disintegrate. This other large cell, this one that again has maintained most of the cytoplasm, is going to be the egg that is going to eventually move into the fertilization process. So meiosis 2 actually doesn't start until the female hits puberty, and then it actually pauses again and finishes when that egg is fertilized. So here we have another picture. This is the picture from your book that shows how the polar body works and how the chromosomes are separated. So it also follows the alleles that would be found on these chromosomes. So if we look at these chromosomes that are found in this first cell, they're telling us that on one homologous chromosome we have a big A allele, and on the other we have the little a allele. So during the first round of meiosis 1, those homologous chromosomes are separated. And in separating that, we see that we have the large A allele going to one cell and the small a allele going to the other cell. You can also see the representation of that cytoplasm. Almost all of the cytoplasm goes to that primary oocyte, and almost none of it goes to that polar body. Then the cell goes through meiosis 2, and we're left with an egg that has proper number of chromosomes for fertilization. And then they have diagrammed those polar bodies. Now, one thing that's pretty cool that you can do with your cells is basically assay them or do an experiment on them, look at their genetic information to see what that human egg has in it. So that primary polar body actually is going to get one of the alleles, while the actual human egg is going to get the other one. So if you know that a mother is a carrier for a disease, you can actually test that first polar body to see what allele it's carrying. And then you know if that egg has the other allele. So this can sort of allow you to see what alleles the child would be getting from this egg. So this process of oogenesis occurs in steps. There's a really nice video on Khan Academy. I put the link on Blackboard that shows how all of this works if you're more interested. You don't necessarily need to go watch it, but if you're interested or confused, you can certainly do that. So meiosis 1 begins very, very early on within the first 12 weeks or the first trimester of a baby's growth and gestation. So meiosis 1 actually happens early on and then is arrested or paused even before that baby is born. So the outcome of this is that girls are born with all of their future eggs present, and they're arrested at prophase 1. So they form their synapses, their chiasmata. They are all ready for their crossing over. And then once the individual reaches puberty, which would be about here, as the eggs are released and are getting ready to be released and then released, they enter back into meiosis and they complete phases from meiosis 1 to meiosis 2, and then that egg pauses again. Now, if that egg is fertilized by a sperm, then that fertilized egg is actually going to complete meiosis 2, and at that point is a fertilized zygote. So a zygote is just a fancy term for saying a fertilized egg. So remember that we said that crossing over had a couple of purposes. One of those purposes is to line up all of your homologous chromosomes correctly in order to make sure that they are partitioned into the daughter cells at the end of meiosis correctly. Now, the outcome of that is that you get variation. You get that exchange between the chromosomes. But the reason that it's so important is that cells that contain the correct number of chromosomes ensure the survival of the organism. If you end up with a gamete without the correct number of chromosomes, you're going to have problems for the organism. most cells that don't have the correct number of chromosomes are not going to lead to viable offspring. So because of that, meiosis and those chromosome checkpoints that make sure that those chromosomes are lined up are very tightly controlled. So here is a summary of those meiotic checkpoints. Down here on the bottom, we have checkpoints that are the same as the checkpoints in mitosis. We've got that start checkpoint, which is the one that tells the cell that it's ready to begin that DNA synthesis, which is basically the start of preparation for mitosis. And then we have that replication checkpoint that's going to make sure that all the DNA is replicated, that there's no damages. Then once we enter meiosis, we have a couple of checkpoints that ensure that the chromosomes are going to be partitioned correctly into their daughter cells. So that first checkpoint is the synapsis checkpoint. That checkpoint is going to make sure that we've got all of our homologous chromosomes paired up, that synapsis has occurred, that crossing over has occurred, so that they're all held together when they begin to move towards the metaphase plate. That is tied to that chiasmata checkpoint, which is where the cell is going to check to make sure that exchange has actually occurred. And then we have the spindle attachment checkpoint. So that attachment checkpoint is going to make sure that microtubules have been successfully attached to the kinetochore. Now, this is to make sure that those chromosomes are correctly pulled apart, that homologous chromosomes are separated correctly, which means that you're going to get 2 sister chromatids in each cell, but only 1 homologous chromosome. But sometimes that checkpoint fails and microtubules don't form correctly. Those spindles don't form and the problem doesn't get found out, and the cell proceeds past that checkpoint. When those spindle fibers don't attach correctly, those chromosomes are not going to be separated correctly, which leads to something called non-disjunction. So disjunction means to break apart a junction, break apart an association. So non-disjunction means those chromosomes don't pull apart correctly. So if you look here, we've got only 1 spindle fiber attached to that chromosome. Now, if you look at the other side, we've got a whole bunch. So that 1 microtubule is not going to be enough to put enough strength on the chromosomes on that side to be able to pull it over. They are telling us that we've got a heterozygous individual here where 1 homologous chromosome has a big A allele, the other has a little A allele. Now, they're doing that so we can sort of link genes to this chromosome movement. So as anaphase occurs and pulls apart those chromosomes, you can see that these chromosomes down on the bottom have been pulled apart correctly. Their homologs have been separated. But up here on the top, the homologs haven't been separated. And so that leads to a problem in the next generation of cells. In this cell on the left, once meiosis 1 is finished, there's no blue chromosome there. There's no A alleles for that chromosome. The other cell, the other half, has received both copies of that chromosome. So this cell that is missing a chromosome is called null isomic. Okay, so somy or somic refers to number of chromosomes. Null means none. So it means that it's missing 1 chromosome, 1 homolog. The other cell is disomic, which means that it has 2 copies of that chromosome. So this problem continues when the cell goes through meiosis 1. So it's not actually showing both of the cells going through, meiosis 2, not showing both cells going through meiosis 2, but it is showing the once you separate your go through meiosis too. It's sort of showing like one of each of the cells that results. So in your nullosomic cell over there, you separate the chromatids, and again, you end up with a cell that is haploid, but is also missing that blue chromosome. In the other cell, when these over here are pulled apart, you end up with both sister chromatids in one cell. So that particular cell now has 2 copies of that chromosome. Again, we end up with a nullosomic cell and a disomic cell because one has no copies of that chromosome. The other has 2 copies of that chromosome. So this is if the non-disjunction occurs in meiosis 1. If the non-disjunction occurs in meiosis 2, that should say 2 there, we get a slightly different situation. So I wanted to draw that for you here. So we've got our cell that is going to undergo meiosis. I'm just going to draw one set of chromosomes here to make it easy. I have drawn the chiasmata, the crossing over that has occurred. And if we just follow this cell through meiosis 1, we will see that each of the cells gets 1 homologous chromosome, 2 sister chromatids. Now, if both of those undergo meiosis 2, and in the cell on the left, we're going to say that it occurs normally, meiosis 2 occurs normally. We're going to separate those chromosomes. So now we have a haploid cell that has 1 copy of each type of chromosome. Now let's say that in that other cell, we have a non-disjunction event. So in this cell, those microtubules don't form correctly. We'll say that they don't form on this side over here. And the result of that is that those sister chromatids are not pulled apart correctly. So again, we end up with a nullosomic and a disomic cell. Now the difference here is that when the non-disjunction occurs in meiosis 1, you have no normal gametes. All of your gametes are either missing or have extra chromosomes. Here, though, in this situation, you actually get 2 cells that are normal. So those cells could potentially go on to produce normal offspring. And you get 2 cells that are abnormal or missing or have gained an extra chromosome. When one of these cells on the left, the normal cell, is fertilized, you get a normal individual. When one of these cells on the right is fertilized, you either end up with a monosomy, which means the zygote is missing a chromosome because this cell doesn't have that red chromosome, but when it is fertilized by a sperm, it will have that chromosome. So I'll have only 1 copy of the homolog. So it would look like, let me get my red back. It would look like this when it's fertilized if that chromosome came from the sperm. In the other situation, if you fertilize that disomic cell, you get a zygote that is going to have 3 copies. It's going to have those 2 copies from the egg plus a 3rd copy from the sperm. So I put up all these definitions here. Nullosomic and disomic refer to the unfertilized sex cell, the sperm or the egg. Monosomy and trisomy refer to the fertilized zygote or that fertilized egg, but they all refer to number of chromosomes. So the resulting fetus from one of those nullosomic or disomic cells is going to produce a chromosome abnormality. So the baby is going to be born with some sort of incorrect chromosome complement. So some stats here to show you how often this occurs. 6% of all conceptions or pregnancies contain abnormal chromosome numbers, which is quite small, so this isn't extremely common. 20% of all conceptions terminate in spontaneous abortion. That usually means that the the fetus is lost even before the woman knows that she is pregnant, but not necessarily. Of those spontaneous abortions, 30% contain chromosome imbalances. So a large percentage of those spontaneous abortions are showing these chromosome imbalances, which suggests that individuals can't survive without the correct amount of chromosomes. Most of those spontaneously aborted fetuses are trisomic for one of the chromosomes, which means there's an extra copy found. And actually, I misspoke. Spontaneous abortion means that basically the mother has a miscarriage, not necessarily that she doesn't know about it yet, doesn't know about the pregnancy. So of the fetuses that they have surveyed, most of them, or a significant number of them, are trisomic. They very rarely find monosomies. Now, this is because it probably is indicating that those cells that are monosomic are never even developing to the point where a woman would recognize that she is pregnant. So that suggests that monosomies are a big problem. Now, this makes sense because in a monosomy, you're actually missing a chromosome. So you're missing a portion of your genetic information. Trisomies tend to be a little bit more common because you have all the information there. You just have extra information. And people tend to do quote unquote better, I use that word lightly, better with extra information than with missing information. So the most common trisomy is trisomy 21. This is also known as Down syndrome. So most of you are probably familiar with Down syndrome. Down syndrome occurs as the result of a non-disjunction of chromosome 21. So that means during meiosis, homologous chromosomes are not pulled apart correctly. Now, chromosome 21 is a really small chromosome, and it normally only forms one crossing over event. So the crossing over, or the chiasmata, is important because that's what holds those chromosomes together and moves them together to the metaphase plate. If you don't have any form, the homologous chromosomes are going to line up in their own places, not associated with one another. If you have only one, such as in the case of chromosome 21, because it's really small, it's not really strong enough to keep those chromosomes together. So if you have a chiasmata in the center of chromosome 21, it's actually better at holding that chromosome together. But if the chiasmata occurs towards the end of the chromosome, that chiasmata is not as strong, doesn't keep the chromosomes together, and so they don't move to the metaphase plate together, which means that they're going to be improperly separated by the end of meiosis. So that non-disjunction situation for Down syndrome is the most common cause of trisomy 21. But there is another form. This is often called familial Down syndrome. And this actually occurs by a different process. In familial Down syndrome, this is actually a Down syndrome that tends to occur in families, whereas the non-disjunction form does not tend to run in families. It's a problem that occurs in the egg after the individual is born, and it's basically a problem in the process of meiosis, not in the hereditary information. But in familial Down syndrome, it actually occurs as something called a translocation. So a translocation is where a piece of one chromosome breaks off. So in this case, it's this blue part of chromosome 21. And in an attempt to fix the situation, the cell attaches it to another one. For some reason, it's often attached to chromosome 14 when this happens. And so what happens is you have your 2 copies of chromosome 21 plus an extra copy that's attached to chromosome 14. And so even though you have the proper number of chromosome 21s, you have a piece of chromosome 21 that's extra. And so you still are trisomic for chromosome 21 because you have 3 times the information there. So trisomy 21 is almost always a failure to cross over in the mother's egg production. And incidence of trisomy 21 increases with maternal age. The incidence of other trisomies, 16 and 18, which are the other most common trisomies, also increase with maternal age, but not in exactly the same pattern. They don't really know why trisomy 21 increases with maternal age. They think that it is related to the age of the eggs. So if you remember, I said that sperm is produced continuously throughout a male's lifetime. So basically, when a sperm fertilizes an egg, it's pretty fresh. It was just newly made. But those eggs from the mother were produced when she was born. And then have not continued their meiosis until fertilization. So you can see that for trisomy 21, once you hit about 30, well, actually, it begins to spike really more between around 35 to 40. You start to see a spike. And they think that this is because those eggs have gotten old. They've been sort of arrested in meiosis, and that maybe during that time there's potentially an increase in or an accumulation of problems in the microtubules, or checkpoints begin to stop working, something like that. There might also be some environmental factor that is perhaps affecting the increased incidence of non-disjunction. But right now, they don't really know. And then, of course, you see that trisomy 16, that spikes much earlier. So there's very likely something different, potentially something different going on with these different trisomies. So let's talk about Down syndrome just real quick. So Down syndrome produces a specific set of characteristics. That's what the word syndrome means. It means a very specific set of characteristics that are associated with the disease. So this is essentially a phenotype from this disease. There's a picture down here that shows is just an individual with Down syndrome. They have very distinctive facial features. They're often shorter individuals. They tend to have physical and mental disabilities. There's an increased risk of various diseases, such as heart disease and leukemia and Alzheimer's disease. One thing that I wanted to point out is this notation here regarding Down syndrome. So this 4721 plus is just a way to indicate what the chromosome makeup is, and we'll see this a little bit later as well. This is just saying that instead of having 46 chromosomes, this individual has 47. And then that 21 plus is saying that extra chromosome is an extra 21. So you could also see a minus that would indicate that there's a chromosome missing, or you might have more than one extra chromosome or missing more than one chromosome. So that first number just indicates total chromosomes. Second number indicates what's missing or extra. So there are a couple of other aneuploidies. 16 was shown on the previous slide. Trisomy 21 tends to be the most common because it is one of the smallest chromosomes. Now this is important because it's a smaller chromosome, it encodes less genes. That means if you have extra or missing on a small chromosome, you're going to be affecting less genetic information. So these aneuploidies tend to occur more frequently on the smaller chromosomes. There are only 2 other trisomies that tend to survive to term. This is trisomy 13 and trisomy 18. I've got some syndrome phenotypes for trisomy 13, which is called Patau syndrome. The trisomy 18 is Edwards syndrome. There are no known monosomies that survive to term. Actually, there are no known autosomal monosomies that survive to term. That means that the monosomy is occurring for one of the autosomes. Now, this is because, again, you're missing genetic information. 2 exceptions to this are monosomies and trisomies for the sex chromosomes. So another relatively common syndrome is called Turner syndrome. And Turner syndrome is actually where an individual has an extra X chromosome. So normally a female would have 1 X and 1 Y, but in Turner syndrome, the individual has 2 X's and 1 Y. The only known monosomy is called Klinefelter's syndrome, and this is a monosomy for the sex chromosome. So this is an individual that is actually just an X chromosome. I'm sorry, I got it. Turner syndrome is your X only. Turner syndrome is your monosomy. And Klinefelter's is more of a disomy. So in Klinefelter's, males would be XY, but in Klinefelter's syndrome, they have 2 X's instead of 1 X. So that Turner syndrome is actually reasonably common for human aneuploidy, but it occurs on the sex chromosome. And we'll talk about why these are more common once we hit chapter 7 and actually start talking about those sex chromosomes. So the reason that trisomy 21 causes Down syndrome is something called a Down syndrome critical region. Now, this Down syndrome critical region contains something called dosage-sensitive genes. This means that if you have more copies of the gene, you're going to get more expression of that gene, and it's going to have an effect on the individual. And this happens on chromosome 21 because there is this Down syndrome critical region that contains a lot of dosage-sensitive genes. So some genes aren't as affected by being expressed more. It doesn't affect the phenotype of an individual. But on chromosome 21, we have this small region where we have some of these dosage-affected genes. So that means that the genes are being expressed more frequently than they should be if you only had 2 copies. And that's causing a phenotypic effect. So chromosome 13 and 18 also have distinct features because of that dosage-sensitive gene. There is one sort of quote-unquote good dosage-sensitive gene on chromosome 21. Individuals with Down syndrome tend to have less tumors. So things like lung cancer, melanoma occur less frequently in individuals with Down syndrome. So this is due to an overexpression of a gene in the Down syndrome critical region that actually blocks cancer formation. So that's an example of a dosage-sensitive gene that actually is beneficial to the individual.